Hi, good afternoon, everyone. This is Tyann Nguyen from CDC's Outbreak Response and Prevention Branch. Thank you, everyone, for joining us this afternoon. And we've heard quite a lot of interest from different groups who want to learn more about how different agencies use the NCBI pathogen detection pipeline and how it integrates into their current epidemiologic workflow. So today, we will hear from 3 epidemiologists from 3 different agencies and their experiences using the NCBI PDP. First up will be Rashida Hassan from CDC. Next, Cassandra Allen will share her experience from Washington Department of Health. And finally, Paula Pinnell-Huth and Amy Robbins will be sharing their perspective on how they've been using NCBI PDP in their state. Following these presentations, we will then open up the webinar to any questions from participants. So now, Christy or Rodell, were there any housekeeping pieces that you wanted to talk about? Maybe we should have started there first, but.
No, good afternoon, morning, everyone. This is Christy Governor with APHL. I want to remind folks that when we do open up the Q&A portion of the webinar, to please use the Q&A box located on the either bottom of your screen that says Q&A. Please type your question there, and we will, Rodell will be moderating those questions for the panelists. Thank you.
Thanks, Christy. So now let's go ahead and get started. The first presentation will be from Rashida Hassan.
Thanks, Tyann. So I'm just going to start with a little overview, since I'm the first presenter today, about what the NCBI Pathogen Detection Project is, just in case you are not aware. But it's a centralized system that integrates sequence information for bacterial pathogens that are collected from food, environmental isolates, and human patients. A lot of different agencies submit the sequencing data to NCBI, which then analyzes the sequences to identify closely related sequences and SNP clusters. The NCBI Pathogen Detection Isolate Browser is a web-based portal that integrates available information on the isolates that are submitted, any sort of metadata that's submitted with that upload, with the SNP cluster information. And the web link for that browser is listed here if you've not seen it before. The current focus for the pathogen detection project is on pathogens that cause the largest burden of human illness: Campylobacter, E. coli, and Shigella, Listeria, and Salmonella. They have added about 20 other pathogens, with many more that are expected to follow. And routine submissions to NCBI come from state laboratories, CDC, FDA, USDA, FSIS, and Public Health England, although additional countries and institutions may submit sequences as well as needed. This is just a schematic kind of representing all the different data flow processes for the pathogen detection pipeline, various submitting institutions in the US and in other countries that get submitted to this portal, and that can then be used to generate various SNP cluster reports, which we'll go through next.
Oh, Rashida,
so
I'm sorry to bother you. We can't see your slides advancing. Is it possible for you to put it in presentation mode?
It is in presentation mode.
Oh, okay. Yeah, the slides.
Are you just still seeing? You're still seeing the title slide?
Exactly.
I don't know if you have any ideas.
Now we do see your presentation. It's in presenter mode on our screen.
Change it to what? No, do you guys see a different slide now?
Yep, now we see your portal. Perfect.
Perfect. Sorry about that, guys. You didn't miss a whole lot on the other slides, and I will make these available afterwards if you guys want to use them as a reference. So this is just a schematic on how data flows. And then I'm going to talk really briefly about how we use the web portal for NCBI here at CDC. So usually our main focus when we're looking at NCBI is during active multi-state cluster investigations, and our objective is typically to determine relatedness between isolates that are included in a multi-state cluster, and potentially to see if there's additional isolates that are related to our cluster that haven't been included in our cluster code. And this is typically just if we would like to get more of a real-time analysis compared to waiting for PulseNet to add sequences. They have a pretty big workflow, and oftentimes they'll wait until we have a number of additional sequences to add to a PulseNet analysis before adding them. So we might look in here so that we can get a little bit more timely updates if we know that we're waiting on particular sequences from particular states. I will mention this later, but I'll mention it now too. We do always verify these results with PulseNet. Sometimes because they're using different pipelines, the results will be a little bit different. So the first thing that we would do is go to the website that I mentioned earlier, the isolate browser, and you can either click on Find Isolates Now or click on your particular pathogen of interest to search for isolates that you're looking for. Since we're usually using this for active multi-state cluster investigations, we're looking for particular isolates of interest. So we will take the WGS IDs we have for isolates included in our cluster, and we will copy and paste them into a search box. Usually the easiest way for us to do that is to pull them up into the subject line list. You can just copy the WGS ID column. If you select that column and just hit Control-C, it'll copy all those IDs. And then you can go into the browser, paste them into that box. It'll automatically format them just with separated by a space, and it will search for all of those isolates, and then you can just click the search button. So this is typically what will pop up, something similar to this once you hit search. And the information found here will be in this box. So it'll say that it found 11 matched isolates based on the ones that you pasted into your box. So if you typed in perhaps 15 isolates and it only found 11, it just means that those additional sequences haven't made it into the pipeline yet. They're not available in the browser. So here we see we have 11 isolates that came up, 7 clinical, 4 environmental, and it's saying that there are 73 total isolates in the SNP cluster. So NCBI defines their SNP clusters based on a 50 SNP window, which is usually much larger than we're looking at. So we'll keep that in mind, but it's saying that these isolates are part of this larger SNP cluster that includes 73 isolates. The minimal SNP difference within the isolates within the search is listed there as well. And we'll talk about the different SNP differences that NCBI uses in just a moment. But once this pops up, you'll see a little SNP cluster ID there on the left. And if you click that, it'll pop up with the tree for all the isolates that you've selected. So once you click that, you'll see something similar to this. And you'll see in the actual SNP tree, all of the isolates you searched for have been selected and they're displayed in red. All of the blue isolates were not part of your search. You can add any additional isolates you want to as part of your selection by left-clicking them and hitting select, or checking the box in the table above the screen there. So if you pop up, you can just hit the little select button and it'll add it to your selection, or clicking the box up there. On the left, you'll see a table of all the different isolates. In the top, you'll see, it'll say distance between selected isolates. It'll give you a min, a max, and an average SNP range. And then here is the difference between the min same and the min difference that NCBI uses. So the min same is the minimum SNP difference within isolates of the same source type, and the min difference is the minimum SNP difference across different isolation types. So it's going to tell you the min same would be, the closest. If you're looking at a clinical isolate, the closest clinical isolate is going to be, for example, in that top one would be 17 SNPs away. And the min difference would be the closest isolate of an environmental or food isolate if you're looking at a clinical. So just keep that in mind. It's not super intuitive when you look at those different classifications, but that's what that means. And so you'll see the minimum min diff range on that table on the left as well. So if you click on this neighbors button, this is a really handy tool. It'll actually select all isolates that fall within a designated SNP difference of your originally selected isolate. So I searched for these isolates that were part of a multi-state cluster. Perhaps I want to see, are there other isolates in this tree that weren't included in our search that are within that same SNP range? So if you click that button, it's going to pop up with a box and going to ask you to put in your SNP distance range. So this is saying I want to add in any additional isolates that fall within, say, 10 SNPs of my selected isolates. You can select whatever distance this is based on, the range for your particular cluster. Once you hit the add in your number, you can hit the add button, and it'll add any of the isolates that you've added to your selection there in the tree. So it'll highlight them in red and add them to your table. You can also change the information that's displayed on this table above. So you can add resistance information, pHG information, et cetera. This is all dependent on what sort of metadata is uploaded to NCBI. So if that information is not available, it won't show anything when you add those columns, but you can edit whatever sort of columns you want to be displayed here. If you click that button, it'll give you all of the different options. So on the left here, if you click the little minus signs, it'll remove those columns from your list. And on the right, if you click these addition signs, it'll add them to your list. So you can customize the table based on what you want to look at, and just hit OK, and it'll reformat it and take you back to this main screen. You can also filter your isolates by time or isolation type. So if you click the little arrow on the filter button, it'll pop up with a window like this. And you can go to the little source information, and you can check whatever source isolates you want to filter by. So if you only want to look at clinical isolates, or if you only want to look at certain environmental or food isolates, you can select those. And then on this little timeline here, you can filter by time by selecting the timeline on this map here, and it'll filter down your search criteria. I will make a note that the dates included in the NCBI clusters are typically upload dates to NCBI, not often reflective of the actual isolation date for that sample. We have a lot of agencies in particular that have been uploading a lot of older data to NCBI, so it might come up with a newer date, but it might be an isolate from a couple of years ago. So we always cross-reference the actual dates in the system with our own information in Cedric or in PulseNet. So once you're done here, you can just hit the little arrow again on the filter button, and it'll take you back to this main screen. If you want to export any of this information, you can click the export button at the top of the tree here, and it'll export an image of your tree. You can select what sort of file type you want to export it as. And then you can also download a line list of all the different selected isolates, and it'll download whatever sort of columns you selected earlier if you wanted to customize your line list, and it'll just export it as a CSV file for you. Another nifty feature here is the watch feature. So again, this is something we might use for multi-state clusters. If we want to set an alert for any new isolates that are uploaded that fall within a particular SNP range of our cluster, we could set a watch for it. So if you click that button, and you will have to have an NCBI account for this, and I'll talk about that in a minute, it'll pop up with this window. You can create a name for your alert and designate the SNP difference that you're interested in detecting new isolates within. You can save the alert, and then it'll send you an email notification when you have new isolates that fall within your SNP cluster. So the other alternative is if you've pasted your isolates into the search box, sometimes it'll pop up with this message: no hits found, would you like to refine your query? So this just means that the sequences haven't been uploaded or analyzed yet, or they have not met NCBI's quality checks, which are different than the quality checks we have for PulseNet. But there is often a delay. So if there were sequences that were uploaded to NCBI, it might take about a week for them to actually show up in this isolate portal search button. So oftentimes if we're waiting for new isolates, we might go and search regularly to see if new isolates have been uploaded, but this sometimes comes up as well. So I just wanted to mention real quick some queries and searches. You can do customizable searches in NCBI. These are just some of the different variables you can search on. You can search on organism. You can search just for new isolates. You can search within that min-diff and min-same ranges that we talked about. So if you're interested in looking at, are there any non-clinical isolates that fall within a certain SNP range of a clinical isolate, you can do a search for that. These are just some of the typical variables you might want to search on, and the different search terms that you would use for that. If you ever forget how to search for those, if you hover on in the main isolate browser window, if you hover over the variable name, it'll tell you what the search name is. So you can use tax group underscore name with the search name there if you actually want to search by a particular organism. So just because you're not expected to memorize all of these, but you can always go through here and determine your own search criteria. So this is just an example. If you wanted to do a search here, tax group name, Salmonella enterica, and add MIN-DIF 0 to 3 with location USA, new, and then epitype clinical. So this is going to search for any new Salmonella clinical isolates from the US that are less than 4 SNPs away from a food or environmental isolate. So this would be a pretty broad search. You can also save these queries in NCBI. Again, you need an account for that, and it'll send you regular email updates if any isolates match your search criteria. I would just recommend being particularly cautious about this. You can very quickly get a lot of searches that end up in your email box. We have colleagues here who have done that for searches that we've been interested in here at CDC. It ends up being quite a lengthy list. So I would just recommend being a little cautious in what sort of information you're interested in searching for. So you would just enter your search criteria here in the search bar, and you can hit search or save if you actually want to save your. So like I mentioned, these watch and save search options are only available if you create an NCBI account. If you don't already have one, it's free to create one. It's very easy. You can, if you don't already have one, you can go to this login button. It'll pop up with a window like here, and you can just hit the new here, sign up for your account. Just a couple of caveats about the NCBI pathogen detector pipeline. Like I mentioned before, all of the analyses from NCBI must be confirmed by PulseNet. We do this for all of our multi-state clusters. The pipeline used for NCBI is different than what PulseNet uses, so we always verify the information with PulseNet. Typically, they correlate pretty well, but sometimes the actual differences that we're looking at are notable for our cluster. So NCBI does use SNP analyses, PulseNet uses MLST, whole genome and core genome MLST, and H2SNP as needed. So those interpretations tend to be a little bit different. We are predicting that using this pipeline is going to be most useful during this period of WGS transition until we have PulseNet allele codes. After the transition, we're predicting that we're going to be using this mostly to supplement information that we have in PulseNet. We're going to be doing cluster detection and case finding based on these allele codes once we have them in PulseNet. But we can use NCBI to find matches in other countries, to look at non-PulseNet pathogens, and things like that. I mentioned before, but there is a delay in seeing some of the information into the isolate browser in NCBI. It can take up to a week. So just because you know that a sequence has been uploaded doesn't necessarily mean it's going to be available immediately to query in their browser. NCBI does also have their own guidelines for what can be considered good quality and what would pass its quality metrics. So some of the sequences that have been uploaded might be rejected by NCBI, even if they have passed PulseNet quality checks. So if you're not seeing it in that browser and it's been over a week, it may be because it didn't pass the quality metrics. Some of the analyses for resistance, especially in older isolates, might be out of date. So we do recommend, if you want confirmation of any of the information that's in NCBI, to contact NARM for the most up-to-date information for those. And then, like I mentioned, the NCBI cluster range is 56. So there's many things that are in their trees that we would not consider to be closely related. So it's always important to double check the isolates within that SNP cluster that are of interest to you, and to double check the alerts for your saved queries. So if you're setting these search criteria, if you're not specifying the SNP range, it might give you anything that's falling within this NCBI's 50 SNP range, which could be a lot of isolates and a lot of isolates that are not really related and aren't of interest to you. And like I said, just be careful because a lot of the saved searches could result in a lot of email notifications into your inbox. One other caveat PulseNet wanted us to mention is that Vibrio cluster detection by whole genome sequencing is currently based on NCBI's SNP analysis, since there is no other schema developed. So just a very brief overview from CDC's end, and I believe we're doing questions at the end.
So next we'll turn the presentation over to Cassandra Allen from Washington State Department of Health, and she will be sharing her experience with the NCBI PDP in Washington.
All right, thank you. And hopefully you are seeing my screen now. So I am just going to do a live demo, basically showing you what Rashida showed you on the slides of how that actually works and looks in the system live. So my first thing I want to mention is that I find that the pathogen detection portal works much better in Chrome than any other browser. So I would highly recommend avoiding, especially Internet Explorer. And my highly technical way of finding it is to start typing in Google, and it usually will be the first search result right there. And once you get to this page, you click on the Find Isolates Now, and that will take you into the browser. And as Rashida mentioned, it's very helpful if you're going to be doing any work in here to go ahead and get a free account. So you can see that I'm logged into my account here. And when you create your account, whatever email address you use to create your account is the email address that will get any notifications for safe searches and watched isolates. So make sure you use the email that you want to keep using. So I'm going to go one step back here out of the isolate browser, and I want to highlight this help link right here. So this is a very new help document. The folks at NCBI have been really responsive to making updates and then getting the help documentation out there. So this is pretty extensive help documentation that I would highly recommend searching through as you get used to using the pathogen detection portal. And one thing I want to highlight on here is, sorry about the scrolling. There we go. Available data field. So as Rashida mentioned, the variable name that you see displayed on the columns is not necessarily what it's called within the system when you're doing queries. So I would recommend, if you're going to be doing queries, to print out this little table right here from the help document and stick it on your desk, because it's much easier to find all your variable names that way. So we will go back into the portal. So there's a lot of different ways to look up data. So what I want to do is search for a range of PNUSA numbers. So those are found in the strain column here. You can see it says to search, use strain. So we're going to call it strain. And then I'm actually going to put in a range of numbers. Using the square brackets, and make sure you use capitals when you're using functions here, like the to or any other and or search functions. So this brought us all of the PNUSA numbers that fall in that range. And over here, this is a really helpful area to look at. So it shows you that there were 45 search results that you found. So you can, if you want to download these, you can just click download here. And it'll pop up, and here's the list of all the isolates that matched your search. So then another way to search isolates is if you have an existing Excel list, you can copy and paste into there. And this is where this little box over here gets really handy, because we can see that that time we brought up 9,007 and 3 isolates, which is clearly not what we were expecting. So if you'll notice, when I copied and pasted that column, it brought over the strain. So it was adding strain as a search term, which gave us all those extra isolates. So if you delete that, then we go back down to 45. So another way that you can search in here is by date. And I noticed this as I was practicing this yesterday. So in the little hover here, it says use target creation date. If we go back to the help document over here, you will see that it has actually been changed and it's just called creation date. So that little hover is not correct anymore at the moment. So we're going to search by creation date, not target creation date. And we're going to do a range of years. So always use a colon in your search. And then we'll do 2018 to 2019. So this gives us all of the isolates that have been uploaded in 2018 or 2019. You can see that it goes up to 512. If you click on the column header, it sorts by that column. So you can see now our oldest isolate is 1/2/2018. So we're seeing what we expect to see, 2018 and 2019 isolates. You can use this option here to filter down. You can also use the filters over here. And it's as expected, we're only seeing 2018, 2019. And you can choose only isolates that say animal as the source. So there's a lot of different options for how you search and filter things down. You can also do, if you want to filter down more in creation date, look at everything with a creation date of May 1st, 2019. So just to give you some idea of scale, there were 459 samples just from that one single day. So there's a whole lot of data available in here. So the next thing I want to do is combine some search terms to actually answer a question. So my question is, I want to see all Washington environmental isolates that have a clinical sample within 5 SNPs since 1/1/2018. So again, I'm going to look for Washington environmental isolates that have a clinical sample within 5 SNPs since 1/1/2018. So I'm just going to go step by step. So our first thing is we want to filter down by location. So geoloc name is, you can see there, geoloc name is our search term. And I'm just going to put in Washington. And the clinical isolates do not have a state attached to them. Typically only you'll see environmental isolates that are down to the state level. The PulseNet organisms that the states upload just go with location of USA. So we can see that that filtered down and we have just under 5,000 isolates. So the next piece is we want to add our creation date. So creation date 2018 to, and there doesn't seem to be a greater than function in the search term. So if you want to do a date moving forward in time, you can use the asterisk, which is the wildcard function. So earliest of 2018 to whatever. So we'll search that, and so that drops us down from 5,000 isolates to just over 1,000. I always like to make sure that as much as I can check and ensure that searches are doing what I expected, sometimes you will get unexpected results. So then we want to do clinical samples. We want to find isolates that have a clinical sample within 5 SNPs. So Rashida did a good job explaining what these min same and min difference columns are. So in NCBI, there are only 2 types of isolates. There's clinical and there's environmental/other. So the environmental/other covers a lot. It's pretty broad. That'll be food isolates, animal isolates, and then clinical is supposed to be only human clinical isolates. So in this instance, you can see this raw milk isolate, the first one here. It's an environmental isolate, min same. So that means that there's another environmental isolate. The next closest environmental isolate is 9 SNPs apart. Under min diff, so that would be the nearest clinical isolate, it says NA. That means that there's no clinical isolate nearby. So to write our query, if we want to get, so we want, right now we're looking at environmental isolates. We want ones that are within 5 SNPs of a clinical isolate. So we're going to say, and epi type environmental. So this is just pulling our environmental isolates, and min diff. So the minimum isolate of a different type, which would be clinical, is 0 to 5. If we search that, then we get 132. So right now we found 132 environmental isolates from Washington that have a clinical isolate within 5 SNPs. And we can look at this column here, the min diff. We'll go ahead and sort by that. And you can see there's quite a few isolates. We have a finished pet food isolate. Let's click into the browser. And you can see here's a clinical isolate that is within 0 SNPs. So our search is working as intended and we're finding isolates. And you can see our maximum number here is 5. And you could go and change this and say 15. You can't see that, but it now says 15. And you can see we got a lot more isolates. Again, you can use the filters here. We only want to do ones that say chicken. Now, to note, if you just click on chicken, you're not going to get the ones that say chicken breast. So just be careful what you're looking for and how you're filtering things down. So once I have this query built, that was a lot of work to get it done, so I don't want to have to redo it every time. And that is not where I meant to go. There we go. So we're going to go ahead and save this search. So if I just say save, I'm going to say WA what saving does is it not only saves it, you can now go here and view, here's our search that we just saved. It not only saves the search that you could go click on it and rerun it at any point, but as Rashida mentioned, it actually will then send you an email alert anytime there's a new match that matches this search criteria. So if we get another environmental isolate uploaded that matches closely to a clinical isolate within 5 SNPs, then we'll get an email alert and can go check on that. So the next question is, I want to see all of the milk isolates that have a clinical isolate within 5 SNPs, and let's filter it, restrict it down to USA. So if we just, we'll again go step by step to show what happens with the search. If you just search for milk, you can see we get 600 and 33. If we search, so isolation source here is really the column that we're interested in. So we're going to search isolation source milk. And you can see doing it that way, we only get 120. So we're missing some. So let's try adding the wildcard asterisk on the end. And now instead of just pulling in milk, we're getting these other things that have milk and then modifiers after them, milk, raw bovine. But we're still missing some, so let's put the modifier in front. And now we're getting things like raw retail cow milk. So we're getting a good search of milk now, and we want to restrict to USA. So, and make sure you capitalized. Yo. Only USA. And you can see a lot of these will have the states named in them. So we're looking for environmental isolate. So, and epi type, again, that's the name for this isolation type column. You could type out environmental/other, or you can just start and use the wildcard. And again, we'll use the min diff. And there we go. So these are all US milk isolates that are within 5 SNPs of a clinical isolate. But if we look through here, we see some things that are not exactly what we were targeting for. Coconut milk wasn't really what I was thinking of, neither was raw milk cheese. So if you want to have things excluded, you can use the not. So I realize you can't see that in the X there, but it's just capital N-O-T. So we're going to say not coconut, not cheese. And you can see we're down from 35 to 33, and it got rid of those odd ones. So I'm going to pick on Washington here, and we'll click into this tree. And let's say this is an environmental isolate. The Department of Agriculture just said, we have a new Washington raw milk isolate, and we're interested in seeing if there's any clinical matches that come up that are near this isolate. So what we want to do is we have our isolate highlighted, and you can say watch, name it something meaningful. We're going to say we'll start with 10 and say save. So now if there is ever another isolate that's uploaded that's within 10 SNPs of this isolate that I have highlighted here, then I will get an email about it, and I can click through on that and look at that isolate and see if that's something that I care about, something we need to look at more closely. So that's a really helpful feature, especially for things like listeria. We know that listeria can hang out for years and years in a facility. So we might not even be looking for clusters, like we're traditionally looking for human cases that match each other. But with listeria, if you are watching an environmental isolate, then if you only get one case that comes up down the line matching, that might be something of interest. So the watched isolates feature is really helpful. So I'm going to go back. And from here, you can get to saved searches and watched isolates. And I'm going to pull up a search that I had here just to get us into this tree. And this is one example of a way that you can use the richness of the data in NCBI. for example, if you have these 3 isolates and you had a local outbreak, and these are your 3 isolates, and they had a potluck and they had multiple meats and foods that people brought in. So you're really at the hypothesis generating stage. So you put your isolates in here and you start to look around and go, hey, there's a turkey there, and turkey. I'm seeing a lot of turkey. Let's see how much. You can type in the search there and you can see that this is a very large SNP tree and there's a whole lot of turkey on there. If we search pork, no. Pig, no. Cow, no. All right, so what does this tell us? Does this tell us that it's a turkey outbreak? No, but it is a clue that we can use in the hypothesis generating stage that, hey, maybe we should make sure we're looking and getting information about the source of the turkey and things like that. So this is one of my favorite uses of the NCBI data, is to be able to use it in the hypothesis generating stage of the outbreak. Another example, Rashida mentioned the Salmonella, or Public Health England is also a submitter. Look up an isolate here. And when we look up this isolate, we see that, oh, this is a US isolate, but looking around the tree, we'll highlight all the United Kingdom isolates. Ah, that's interesting. So there's clearly something going on with United Kingdom. And this is a Salmonella Chester. So I went to my favorite epi tool, and anything happen with Salmonella Chester in England? Oh, multinational outbreak of travel-related Salmonella Chester infections in Europe. And I found here that there was travel to Morocco and other European countries. And that is a great starting point if you're looking at this case or this cluster here, that, well, maybe before you start to worry about NHGQs or long questionnaire forms, that travel would be a good thing to look at first. So I'm going to start to wrap up here, but just in summary, the way that we are using the NCBI pathogen detection in Washington is for hypothesis generating. So looking at what clinical matches are out there, what matches from environmental isolates are really a strong source of information in here. Another way that we're using it is in this example that I'm pulling up here. We had an outbreak, and when we put the isolates into NCBI, this is a very large SNP cluster, so it takes it a minute to think about it. And if you have, you can see that there's 7,000 isolates on this SNP tree, so it's a little unwieldy. If you want to see a smaller subset, you can click on the node there and say subtree view. And it brings up a much more manageable tree. So the isolates highlighted in red were all part of an outbreak. And when we put them in NCBI, we saw that there was one that seemed to be lumping in with these isolates, but wasn't part of our outbreak. And actually, if you look temporally, it occurred many months earlier than the other cases. So we were able to go backwards from this PNUSA number using Cedric and found that this isolate here that wasn't part of our outbreak was part of a previous outbreak. And we were able to use that information to jumpstart the investigation, and really it helped solve the outbreak much quicker. So being able to go backwards from the NCBI PNUSA data to the Cedric data to pull isolates and see what previous isolates were related to can be really helpful. So we're also using it, like I mentioned, to track environmental isolates. So if we go to watched isolates, there's quite a few environmental listeria isolates here. We like to keep track on those, knowing if we have one new listeria case, even though these are from many years ago, can be helpful. So we're using it to track environmental isolates. And then one feature that is pretty handy is for if you're looking and waiting for one particular isolate. I'm just made up a PNUSA number there, which hopefully it doesn't find. So it says no hits found, but I really want to know as soon as this one's available. So instead of coming back here and searching every day, you can save that as a search. And we'll just name the search that one. And then as soon as that isolate has been processed through NCBI's pipeline and is available, you'll get an email saying you have a new search result. So that's a really easy way to kind of pre-watch an isolate before it's even in the system. So that is the ways that we're using it in Washington. It's been a really helpful tool. And I think Paula in New York is going to show some more demos of how they're using it in specific outbreak examples.
I am just about to share my screen. And you should be seeing the title slide. Amy Robbins is going to speak first. And yes, we're going to, given the time as well, does this go to 2? Is that what time this goes to?
Correct.
Okay, so we'll just go briefly, and it was really great how the last 2 presenters kind of did the NCBI demo, where we have screenshots and different things. So we may or may not log into the system given the time.
Good afternoon, everyone. This is Amy Robbins. We're going to just give you guys some examples of how we used NCBI in the past, but mostly when we were still also doing PFGE at the same time. So some of these clusters are also with utilizing PFGE results we had first. So the first example is a Typhimurium cluster. And in this cluster, we were notified of cases rather slowly and just did not come up with any kind of common epidemiology amongst the cases. And there's no common foods, no common travel, nothing. And then of course we had the one that came in with no interview. So we decided to look it up and see, well, how could we utilize NCBI to see if these cases are truly related or not. So once they were all in NCBI, and since you guys saw the demonstration, I'll just use these screenshots. You can see on the main screen, this is where I put in all 5 of the PNUSA numbers, split with a space, and I got this result on the search screen. And so what this was saying is that from the 5 that were entered, 2 of them are in one cluster, and the other 2 are in separate clusters, greater than 50 SNPs apart. So really nothing to do with each other, which made a lot of sense considering that we couldn't find anything in common food-wise or anything else. So then when looking at the 2 that were somewhat related, or in the same cluster, at least, by clicking on the hyperlink for the SNP cluster, able to see the tree. And in the tree, we did the subtree view that Cassandra just showed, and we're able to find out that by clicking on the dots in front of the 2 isolates we were interested in, we were able to find that these were 8 SNPs apart. One of these cases was not interviewed, so we didn't have enough information to continue going forward. But after weeks of trying to figure out what did these 5 cases have in common, if we had waited for sequencing results, basically would have found out that we wouldn't have looked at all 5 cases, just those 2 cases. So that was kind of the help that we've been doing a lot with NCBI, is helping us to basically say that we did not have to spend the time to investigate those clusters. So by
So just looking at a few of our data from 2019, we did find this is the important one, that it looks like a number of our Salmonella clusters were not related by whole genome sequencing, about a third of them. And this information was presented at a conference in January, so it was not complete at that point in time, but it does show to us that relying just on whole genome sequencing is going to have fewer, potentially fewer clusters of Salmonella, except maybe not for the endemic patterns. I'll turn it over to Paula.
And so I'm going to highlight an example where we definitely use environmental isolates that were included within our tree to help further our investigation. Again, it was Salmonella typhimurium. And again, back when this all began, we were still utilizing PFGE and whole genome in tandem, with PFGE results getting to us a bit sooner. So from November 2017 to May 2018, New York State Department of Health investigated 10 PFGE matching cases of Salmonella typhimurium in New York State, excluding New York City. This number of cases was not above the baseline for what we normally see with this pattern. And when you looked at the case interviews in totality, they really didn't yield any striking common exposures. In June of 2018, Wadsworth Center completed whole genome on the 10 PFGE matching isolates. At the end of May, four matching cases by whole genome were reported, and then another came in closer to mid-June. And so I'm just kind of clumping them all together. When we looked at those 5 of the 10 cases that were told to be high related 0 to 6 SNPs, there was definitely a kosher type of association. One case had reported a kosher diet. A couple other cases had reported a grocer that was, maybe specializing in kosher products, but that wasn't their exclusive grocery store. Most had multiple types, and just from your general commercial ones. And then when I went into the investigation forms for a couple others, I was able to tease out a couple brands. Like one was for a peanut butter. They had just happened to mention the brand, and it wasn't a brand I was particularly familiar with. So when I looked it up, it was a kosher brand. So kind of linking different pieces of the interviews together, I noticed some sort of association. This was just an example to show how the first 4 whole genome sequencing cases were reported to us, matching cases using the pipeline at Wadsworth Center. And then you'll see the other ones that are, so the ones in the circle were the ones that eventually became part of this outbreak, 0 to 6 SNPs. And you can see how 5 others, 5 of the other cases that we had been looking at when it was PFGE are now, not associated with the outbreak in question. You can see they're 12 to 14 and then 66 to 72. So, again, they may be, clusters of their own, but they weren't part of this new one we were looking into. This is where I went into NCBI, and I have screenshots. I was going to go in, but essentially what I did was I just took a few, I took the isolates we knew, the New York isolates, I put them into the identified isolates now search box, and what came up and I looked for within 0 to 8 SNPs. And I found 6 additional clinical isolates, and they had very recent collection dates, January to April 2018. And I found 6 USDA chicken isolates, again with recent collection dates, December 2017 to April 2018. I'm a big fan of what's called the sniffing tool. It's probably the least technical thing on your computer, but I like it because essentially, even though it's good to export and download your searches, anytime you click on that link again, it'll bring you to what is currently happening and not to what you were looking at way back when. So because this was so new to me back when I was utilizing NCBI, I literally took screenshots of everything because I wanted to make sure I was reading it right and passing it back and forth with the lab, just saying, is this, I'm reading this right? So this is an actual shot from back then, where you can see the red one that's underlined was the one I typed in the search box. And then you can see that there's about 6 environmental isolates listed, and then a variety of clinical isolates. And then from that, what I did was I utilized Cedric to put in those state isolate IDs to find out where those isolates were from that seemed to be in the 0 to 8 SNP range of our cluster, of our cases that we knew about. And I identified cases from, there was an additional case from New York State. They had been analyzed in New York City, so it didn't come across on the lab report from Rodsworth. So we identified an additional New York case, two New York City cases, and then a case from Pennsylvania, Vermont, or Virginia and Maryland. And so I reached out to the epi contacts to see about exposure information on their cases, basically saying kosher ? And when I got the replies, yes. So there were 11 cases, 5 with reported kosher food exposures, 3 with possible, and I say that in the most literal sense, kind of relying on some of our old methods of epi of, well, they, you know, maybe listed a grocer, but didn't specify any foods, or maybe it went to a restaurant, but didn't specify. So it was 3 there, and then 2 with no kosher links identified. 1 case was lost to follow-up. All cases reported any chicken consumption. 8 out of 10 also reported dairy. 6 out of 10 also reported cucumbers, zucchini, or squash. At this point, I reached out. I mean, essentially, when I saw the tree with both the additional clinical and environmental isolates, reached out on the epi side to clinical. For the clinical information, reached out to our environmental health investigators and asked if they could look into those 6 USDA chicken isolates, hoping, again, they had a kosher association, and they did. They were all products of kosher or halal processing facilities. And we all know this like rarely happens for us, that everything is lining up. So it was super exciting. June 20th, I contacted CDC to share our hypothesis that illness was linked to a kosher-related food product, possibly chicken. That's when we decided to re-interview the 11 cases with an HGQ focused on chicken and focused on brands that were implicated by way of the products in the NCBI chart with the brand names to help with recall. When again, 1 case was lost to follow-up, again, refused interview. But the other 10 cases did report exposure to chicken, and those who could call a brand name cited a specific brand of poultry. I would do the demo. We're short on time. So again, this is more at the end of the, as the outbreak is ongoing, more cases were reported. You can see the 1 case highlighted in red. That was one of our cases. And back then there wasn't the neighbor function or the different functions we have now. So it was kind of a hunt and peck method of, you would just click on each node and we wanting to stay within your 0 to 8 SNP range, which is what we had defined. And when you did, it would look something like this, where some are weeded out just because they go across the limit, and it's just as your way of additional case finding. And this cluster is posted on the CDC website. So just in conclusion, it's just using NCBI, we identified a close relation between the cases who reported exposure to poultry brand A and food isolates from brand A establishment. And again, we were able to do this using NCBI and looking at isolates who were within our SNP range that we wanted, and really using the environmental isolates to our advantage, especially when you're dealing with something, you know, like chicken. It really helps to then give you more focus on what type. And just acknowledgements. And we do have a nice demo on the COE website. It's a little bit more of a basic introduction than what was given today, like by Washington. I mean, we were learning today from that, so that was great. There has just been so many new search functions added. But this is, if you're very new to NCBI, this would be very helpful. This is how we learned how to use it. And it goes through the different core features you might be interested in. And that's all.
Thank you, everyone. We're actually ahead of schedule a bit. We scheduled this webinar to go to 2:30, so we'll have about 30 minutes for any questions that people have.
So we did have 2 questions so far. One was on the chat. Let me pull it up real quick, and this would be addressed to Rashida. So someone wanted to know, how does NCBI define new clinical isolates?
So it would depend on how you're defining your search. So in the watched isolates, it's going to send you a notification anytime, immediately once something is uploaded that meets your criteria. So it would be a regular update. So it would depend on how often that an isolate meets your criteria. If you're doing a general search and you're looking for new isolates, I believe it depends on how long it's been since NCBI has run their calculation. So it would depend on the pathogen. So for salmonella, I think it would be probably within the last 24 hours, but for things that don't update as often, it might be longer. So it would be, I think, pathogen specific for that. But for the most common utilization, I would think for that would be for like watched isolate.
Awesome. So our next question, someone wanted to know, and this is probably for Cassandra or Paula or Amy, they wanted to know if you can go back over how to bring up that save button to show how you can save your searches for isolates.
Sure, so this is Cassandra. I can share my screen again for a minute. So, question was how to find the save button.
Yep.
So the save button is right here. I believe the save button only shows up if you have an NCBI account and you're logged in. So if you're not logged into your account, there's no option to save.
Awesome. So our next question, if you're done, someone wanted to know, can you show where you find the subtree option?
Yes, I'll click into a tree here. So so I'm just hovering over this node here, and if I want to see a tree that just has these isolates, you left click once and it'll say subtree view, and then click on that and it'll think for a minute, and then hopefully pull up your subtree. Well, that's thinking. Related to that, there's one new feature that they just added, I think last week, so I haven't had a chance to play with it. But if you have isolates, say you have a pretty large tree and it's a little unwieldy to, say you wanted to compare an isolate from down here with way up there, you can select them both and say common subtree, and it'll just trim your tree down to the lowest common denominator. So it'll get rid of a few of the extra isolates. And this one is still thinking about making your subtree. Sometimes the subtrees are a little finicky. They do usually show up eventually. And there we go. There's our subtree. So just makes it a lot easier. And then if you wanted to click on that and select all leaves, then that gives you an easy way to look over here. And you can say that within this subtree here, they're all within 0 to 9 SNPs.
Awesome, thank you.
Great, thank you. Rodell, are there any other questions?
Not at this moment.
Great. Well, we'll give folks another minute or so to ask any questions. I don't know, Tyann, do you have any other thoughts as we wait for questions?
I don't have anything in specifically, but I feel like as more and more states and local health departments use the NCBI PDP, and you know, it looks a little bit intimidating until you get really into it. We invite you guys to reach out to CDC, or I'll also volunteer Washington, New York, and anyone else, any other states out there that use it on a regular basis. I think from our end, we're more than happy to help answer any questions.
So we did get one, and again, if you have a question, please post to the Q&A box. But we did get one. Can any institution post to NCBI, or is that specific to particular agencies?
Hi, this is Lee Katz. I'm the Senior Bioinformatician in the Infectious Disease Laboratory branch of CDC. Yes, anybody can send their sequences to NCBI. The trick is that you have to come up with your own bioproject and notify NCBI to be monitoring that bioproject, and they will analyze that bioproject for you and bring it in with the rest of the genomes.
Awesome, thank you.
Great, thank you. Are there any other questions?
Nope, not at this moment.
Well, great. You know, I'd like to thank our panelists from Washington, New York, CDC, Tyann, and Rodell for working on today's webinar and presenting your experience with NCBI's pathogen detection portal and how you're using it in your EPI workflows. This webinar is being recorded, so APHL will be sending out information regarding accessing the recorded link.
So we did have another question that came up as a follow-up to the last one. Do they have to have, do the specific institution have to have, do they have to meet the PulseNet criteria requirements to upload to NCBI?
Hi, this is Lee Katz again. That's a tricky question. So just like as an individual or someone outside of PulseNet, like anybody can really upload to this and work with it. But as part of PulseNet, we prefer that people adhere to the PulseNet standards and even notify PulseNet of what your bioproject is so that we can kind of stay associated online too.
The person just added, this is an existing contributor.
As an existing contributor?
Yeah.
So if you're already part of PulseNet, you're part of the MOU and everything, then yes, we would prefer that you kind of notify PulseNet what your bioproject is and adhere to those standards.
Awesome, thank you.
Good question, thank you.
Great. Well, one last shout out for any questions, any remaining questions? If not, then I guess we could close the webinar for today. Again, I'd like to thank all of our panelists for their time and for you all for joining today's webinar. Be on the lookout for the recording, and please let us know if you have any further questions. Rodell, do you have anything last minute questions you want or comments you'd like to say before we end?
No, that is all.
Great. Well, thank you, everyone.
Bye-bye.
Bye-bye. Thanks.